Hello, the students of plus one. In the second lesson of the biological classification, you studied about the definition of classification and then three domains of life, and these are bacteria, eubacteria, archaea, and you have studied till now the different characters of the monera. The monera are the prokaryotes bacteria, and they are prokaryotic. They have no nuclear membrane, they have no mitochondria, plastids, only DNA, circular DNA. And in moneran, they are the photosynthetic autotrophs, heterotrophs you studied, and in moneran there are archaeobacteria. The archaeobacteria are old bacteria, primitive bacteria, methanogens, halophiles, and these are the archaeobacteria. Now then you studied about the bacteria. The bacteria are studied by Leeuwenhoek, and the blue-green algae are blue-green algae having photosynthetic pigment in the near the cell wall. You have studied the before, and then you switch the topic. After monera, there are protista. Protista has, these are eukaryotes. Monera are prokaryotes and protista are eukaryotes. Eukaryotes in protista, you will study many other organisms which are dinoflagellates. Dinoflagellates and diatoms. These are the diatoms. You studied diatoms, and the diatoms are very interesting protista. Diatoms are golden algae, and cell walls having silica made of two halves, like the box of the sweets, two halves, and the two halves can be separated by a very interesting way. The two halves of the diatoms separate by an interesting way of the asexual methods of reproduction. These asexual reproductions are, this is the diagram. So, like the box, it has epitheca, hypotheca. The two epitheca and hypotheca separate. The hypotheca make new epitheca, and epitheca make new hypotheca. By this method, they reproduce by asexual manner. This is called binary fission. It is very interesting. Like the box of the sweets from the market, we take upper lid, lower lid, epitheca, hypotheca. Hypotheca is lower part and epitheca is the upper part. By this method, the diatoms are reproduced by asexual manner, and the diatomous earth in that very ocean. Then we studied dinoflagellates. Dinoflagellates are also prokaryotes, and they have reserve food in the shape of oils and polysaccharides. They are red in color, make red tide, and they have covering with theca, and two type grooves, horizontal grooves, vertical grooves, and two flagella grooves, horizontal flagella and vertical flagella. These are the dinoflagellate. Regarding diagram, you can make this diagram. Yesterday I told you about the diagram of the dinoflagellate. They are single cell and having shell, then interlocking plates, two flagella I told you, autotrophic, photosynthetic, reserve food is polysaccharides. Goniolax, accumulation of goniolax make at the bottom of the sea makes red tide, and it is food for the mussels. Mussels means pila globosa. Mussel is an animal. In Hindi we call sippy. Mussel eats the dinoflagellate. Then eukaryotes. Eukaryotes are the euglenoids. Euglenoid. Next part is euglenoid, and very good diagram. The euglena is there. You can make this diagram from the book. So euglena are the organisms which are Protista, and but it has photosynthetic pigment. Photosynthetic pigment. It has flagella, photosynthetic pigment, nucleus at the base, paramylum bodies as food, and paramylum body. This is paramylum body, and these are the we can say that is chloroplast. These chloroplasts help in photosynthesis. It is animal or plant, though it has both characters, plant characters and animal characters. In euglenoids we study euglena, and then after it is made of slime molds. Slime molds, we will study slime molds, and then Sarcodina. Sarcodina are amoeboid protozoans. Amoeboid protozoans are Sarcodina. They are protozoan protist. They are 4 types. Number 1, Sarcodina, Mastigophora, Sporozoa, ciliata. So Sarcodina, this is Sarcodina, and then Zooflagellata. These are protozoans. Protozoans are Zooflagellata, and the examples of the Zooflagellata is Trypanosoma. Trypanosoma is the organism which causes disease, and Trypanosoma and Sarcodina. In Sarcodina we study amoeba. You know about amoeba. Amoeba is a unicellular organism, and with pseudopodia, locomotion, lobopodia, pseudopodia, and nutrition heterotrophic, reproduction by binary fission like bacteria. Next we have to study ciliata. In ciliata, very good diagram, very good organism. We study paramecium. Paramecium is called slipper animalcule. You study the characters, make the diagram of paramecium, amoeba, and study this topic, and we will study tomorrow.